slideshow and from the beginning. I hope
See it, both of you.
everybody could see this and I am audible to everyone. So as I said just now, we'll be talking about artificial intelligence and we'll especially talk about its application in symbiotic systems. Though I'm trained and I have all uh, most experience with one type of symbiosis studies, that is arbuscular mycorrhizal fungi. But because here my theme was on artificial intelligence, I have just tried to stretch, find the applications where all in symbiosis, and I work with symbiosis a lot, being the vice president of International Symbiosis Society. So I tried to cover the examples wherever I could facilitate the application of artificial intelligence in symbiotic systems. So that is the first slide. And I know because you had previous lectures, I was just seeing your schedule in this one. So we don't need to talk and try understand what are symbiotic associations. But I hope you believe and you understand that it refers to all interactions, all persistent interactions where one partner may live in the body of the other, or it may be interacting through a distance. This is what we include in symbiosis. And then I just want to remove this, just 1 minute. No problems. Let it be like this. So here I have just taken some pictures where I just wanted to show the variation we get in symbiotic associations, and the most common one like mycorrhiza, microbiome you talked about in the morning, coral leaves, lichens. Silvana has already talked about lichens and she is the expert. And at places, maybe we would love to get some suggestions from her that what I'm talking about artificial intelligence with reference to lichens. So I would like to have her comments on that. So we will cover all these symbiosis types when we are talking about artificial intelligence applications. So studying symbiosis or symbionts can be a bit tricky because not all microbes can be cultivated by common microbiological methods. Like, for example, arbuscular mycorrhiza, which is an obligate symbiont, we can not culture as it is the fungi without the host in the laboratory. And if we try to identify this by normal routine methods, the morphological characters of these fungi are not stable, meaning we cannot identify them by the normal morphological characters. We need to go to molecular methods. And in the molecular methods, of course, we have some stability, but we need to develop more methods so that it is much easier to identify. Same way, these microbes, when we're talking of interaction, that include millions of the microbes, they vary in size. They are huge in size very often when we look at them in the marine environment, and they are at very distant locations. Maybe for lichens, you might be studying the lichens from tundra region or maybe the Antarctica. So they vary from different regions, and culturing them together very often gives different results and for different symbionts. And even if we try culturing them, the methods are very lengthy and tedious. So with looking at all these problems, we shifted to high throughput sequencing methods and the technology, and we needed to have advanced data analysis methods. So high throughput sequencing technology gave us huge amount of data, and we needed to shift to the bioinformatic data analysis techniques. So these things developed huge amount of data, and we gradually had to go to bioinformatic methods. And the other area where these symbiotic studies have really flourished or had advanced is the image data. With these symbiotic organisms, you get huge data, maybe as microscopic images or as manual images taken by the videography in the marine environment, or we have huge video data. And we also have remote sensing data. And I know that a lot of studies using this remote sensing data, they are trying to study the lichen distribution. So the image data may be from remote sensing data or manually collected by camera, or maybe microscopic camera has captured that data. That data is also quite advanced and accumulated, and we can derive huge information from this image data. We can use it for automating things. So that was one type of data which is accumulating, and the other type of data which is accumulating or which we have for study of these symbiotic organisms in a particular environment is the metagenomic data. And by now, all of you are very well familiar with metagenome. What is a metagenome? So we have a data from one particular type of environment for this metagenomic process. So say, for example, I have shown you the marine environment here. So what we do in collection of metagenomic data is we'll extract all DNA from the microbial community in this sampled environment. We will determine what genes are available here, and we will assume that one gene refers to one organism. We determine the biodiversity. We try to identify the genes, and then we also compare, and we also try to determine the function, maybe whether this serves our interest, or meaning what a function it may be performing. And in several other studies, we also try to know the phenotype being controlled by these genes. So with this background, I am moving on to artificial intelligence. Until now, what I was trying to focus is that we'll be talking about artificial intelligence with reference to two data types. One is metagenomic data, and the other one is image data. Image data can be from any source, maybe microscopic images, maybe the remote sensing images, or maybe the uh, manual camera images, video images, which you are getting from the marine environment. So what is, yeah, it's better now for me because these pictures were troubling me. So that's better for me. Yes. Yeah. So what is artificial intelligence? Artificial intelligence refers to simulation of human intelligence in machines that are programmed to think like humans, meaning we try to program machines so that like our brain, they can mimic the actions which we want to perform. So artificial intelligence, we try to mimic the activities of human, and we try to get it done by machines. Right. So few questions which we try to answer is, can computer play chess like humans in artificial intelligence? And the answer which came is yes. And in the computers can beat the best humans in chess. The next question which came is, can computer talk like humans? And again, the answer came as yes, but it can perform specific tasks in few languages. The question which came further is, can computer understand speech? Again, the answer is yes, but for very specific domains. Can computers learn and adapt? Same answer, yes. And can computers see? Again, the same answer. So working with machines, we did get success in all these forte, but work is in progress. And we got success, but we have limitations. Like, can computers see? Of course, face recognition is working. So that was about artificial intelligence. And now you know that artificial intelligence or intelligent systems are there in your everyday life. You use that for image processing, you use that for face recognition, you use that for bioinformatics, gene expression, you use them for medicine, or maybe all sort of things, handwriting recognition. Artificial intelligence is everywhere nowadays. So starting with the AI applications in symbiotic systems. In the symbiotic systems, we will be talking about two main type of data. I told you already, metagenomic data or image data. And in the metagenomic data, we will understand how AI application is helping in biodiversity profiling, or maybe the functional annotation, phenotyping of this data, or maybe for the image data, we would understand how these three types of data have deciphered or have helped in knowledge extraction from these studies on the symbiotic organisms. So starting with artificial intelligence once again. So in artificial intelligence, we try to mimic, to try to do the task with the same level of human intelligence, and we try that the computer does our task at the same level of intelligence. We have machine learning, the most popular artificial intelligence method. And what machine learning is the machine learning methods or the techniques, they learn from the data, meaning the algorithms are designed in such a way that whatever data input you are giving, it will learn gradually and make things better, like the humans. They learn from the system. Then we have deep learning, and deep learning loosely functions like human brain. It uses neural network, which is like human brain, which mimics, tries to, it is a loose statement, but it tries to function in the same way as human brain. Then we have computer vision, which mainly is the algorithms here, the machine learning. These are overlapping branches, and the algorithms which it uses mainly deal with the image data. So here we'll be talking about the three artificial intelligence applications or methods. One is machine learning, deep learning, and computer vision. And we'll be seeing how machine learning has helped in metagenomic data analysis, and maybe the deep learning, which has helped both in metagenomic and image data analysis. And we'll talk about the computer vision, where we're talking about automated image classification using computer vision. So machine learning algorithms, I am not going very much into the details, but we just have an introduction, how they work, and then we'll come to their applications. So basically, the machine learning algorithms aim to learn directly from the data. A typical machine learning algorithm requires at least three components. It must have an experience in the form of data, maybe a genomic data or image data, meaning it has a data and it tries to learn from the data what best can be done with this data. Then it has a task to perform, the task in the form of output of the algorithm, and that can be anything ranging from maybe we want to do clustering with this data, or we want to do the classification of the data. The function or the task varies from your aims and objectives. And the third component it has is the objective, which is in the form of performance measurement of the given output. So basically, we have these three components in any machine learning algorithm, and it learns from the data. Then if we come to deep learning, what is deep learning? I have tried to simplify the concept as best as I could do, so that it makes sense. So deep learning is what is a type of, is a family of machine learning methods, and it is based on artificial neural networks and feature learning. So artificial neural network, like the pictures shows you, it works like human brain. And what is happening here is, like the artificial neurons, the data input, meaning each neuron has an input and it produces a single output which can send to the multiple other neurons, very much like this picture, which resembles to our nervous system, brain functioning. And the other feature of deep learning is that we have feature learning here. So what is happening is, in the feature learning is a set of techniques that allows system to automatically discover the representation needed for a feature detection. Meaning, in this, the system itself automatically discovers the feature and the feature needed for the classification or detection in this data. So in deep learning, we apply artificial neural networks, which is based on feature learning. Then we have the third technique, which we thought method in artificial intelligence, which we'll be talking again and again, is computer vision. So what is computer vision? Is a field of computer science that aims to understand and interpret the world through video and image data. Right. So that was about computer vision, and it uses the neural networks, and it uses the other type of network that is called as convoluted network, that is CNN. So it is using machine learning algorithms and the neural networks, that is CNN, for its functioning. And it has found, I told you already, it has found extensive applications in geospatial imagery and marine image data. So that's a small picture which I took from our spores, arbuscular mycorrhizal spores. And I'm showing you this picture, just I wanted to show you that all data which is coming from images to computer, it is just a collection of pixels as binary numbers, and these pixels have a pattern. So machine tries to pick this pattern from a picture. Then what next it does is like this one, which I showed you this picture. With reference to this picture, what will machine do is, like I said, this image of spore is just a collection of pixels and a number which has its own pattern. Then we give these labels, meaning the algorithm is provided, the information is that a certain pattern, and on based of these patterns, class objects are defined. Like for this spore, we'll define a class object that we call as spore. Text it, spore. So what happens next? Whenever in any picture, if we feed it a new picture, and the algorithm begins to identify this pattern in the other images, and wherever it sees this pattern, it will say it is a spore. So this is how it functions. But the most important thing which we must know here is that the importance of annotation. The importance of annotation, meaning we must have labeled data available with us, meaning we have to previously tell the computer, the algorithms, that this spore is going to look like this, and we have to make a class of spores. Then only this will be able to tell us, that it will be able to identify, the algorithm will be able to understand in the real world that this is so the annotated data is very, very important. We must have that for doing this. Then once we have images, lots of things are done with that. So we can use this for classification, that maybe this belongs to the spore, or this belongs to some algae, or maybe object detection. In one image I showed you previously, it would tell you how many spores are present on the basis of this. And it can be semantic segmentation, or maybe the instance segmentation. So it will gradually pick up the different parts of this image and will tell you all information about the image. So you have a training data with which you train the machine, that like these spores would be coming. And then you have the test data. After training the algorithm, you would test your things in the test data, and it gives you quite acceptable result. So machine learning algorithms, we were talking of that, and I hope I could tell you what are basic steps in these algorithms, what are these algorithms doing with the images or metagenomic data. It is doing the same thing. I have one more example with the pictures. After I finish up with this, I'll tell you how machine learning have simplified things and have made things more accurate. Like for example, machine learning algorithms, I'm just telling you about the algorithms first. They are classified into three categories based on the annotated data set. I previously told you to have a labeled data is very, very important in this type of work. So we have three types of learning, or three types of algorithm based on the presence of labeled data. One is supervised algorithm, that is, we have labeled data present, we have available. And this is the supervised algorithm, which are mostly used in microbial studies. Like I said, for my AM spores, I needed to have previously identified spores to give a input for training. So supervised algorithms are usually of two types, classification or regression algorithms. In our microbial studies, we use mostly supervised algorithms, and especially we use the SVMs, NBs, RF, and KNN methods. We will not go into the details of this, but when we talk of the applications, we would know these are the four main supervised algorithm methods of machine learning which are being used for the symbiotic systems or microbial studies. That can be unsupervised algorithms also, meaning we don't have a labeled data available and the algorithm itself first decides the meaning it is mostly used for pre-processing or maybe clustering. The on the basis of the algorithm, it doesn't compare, give a name, it cluster and designs the, so it is more used for pre-processing of data. We have something in between also, that is semi-supervised algorithms, where labeled data is available but is very scarce. So these three types of algorithms exist for machine learning, but I said we usually use supervised algorithms. And in the supervised algorithms, the SVM is the most popular one, which is used for classification, most popular classification algorithm used in ML. And originally, it was designed for binary data where dividing it into two classes is possible, linearly separable data, but gradually it is extended to multi-class data where, like in metagenome, where we have a lot of types of species present in one environment, or maybe non-linearly separable data where the data is very hard to separate into one class, one or two class, or linearly we cannot separate that data. So SVM, I said, is the most popular method. So let's start with metagenomic data first of all. Where in metagenomic data analysis and ML algorithms are coming in between, and how they have helped us to improve our result and performance. So in the metagenomic data analysis, we are mainly talking about the sequence-based metagenomics, and I hope you know by now that in sequence-based metagenomics, we could have sequences from based on marker gene sequencing or maybe whole genome sequencing. In the marker gene sequencing, we use markers maybe 16S rRNA or maybe 18S, depending upon whether it is prokaryote or it's eukaryote. And from one particular environment, we will isolate, we will extract all DNA, we'll amplify this, and then we'll do clustering, classification, we'll make a phylogenetic tree out of this, and then we'll compare and give a name to which particular organisms are present. The method is a bit different in whole genome sequencing, where we do not go for a particular marker gene, rather we sequence the complete genome sequencing, and then we assemble this, we try to predict where the genes are, and we analyze these genes, and then we compare. But if you are going for taxonomic identification, it is the marker gene method is finance-wise, cost-wise also, it's more viable, and getting the accuracy and computer time from all terms, we mostly use marker gene. So it all depends on your purpose, what you are going to do with this metagenomic data. If your idea is to study the function, or maybe the interaction, or maybe the phenotype, you'll go for whole genome data. But we talk more about the marker gene sequencing data. And let's take one example. I have just drawn a data analysis workflow here, that what we do in the data analysis for this sequence data, metagenomic sequence data. So after we have sequenced, we get huge data after PCR amplification, and this data is is fed into the system. I hope you know Chime, and Chime is the most popular platform where we do the metagenomic data analysis. So we'll input the data as a FASTQ or maybe the mapping file as it is to the software, And then the first step here, before we go with this data analysis, is quality check of the samples, decontamination, and sample multiplexing. So the quality check, like it explains here a bit more in details that what pre-processing is required and why it is very important to have this pre-processing. Chime is doing filtering, and it could filter the low quality bases, and we have several other platforms available for this filtering. In sample demultiplexing, what we do is we remove the barcode and primer sequences from this the sequences available, and the rates need to be checked, need to be linked back to the samples from where they have come. To get the accuracy in results, this pre-processing of the data is very, very important, and Chime has different scripts and that could help you to do that. That's not an issue. Then we have the third thing in pre-processing, that is decontamination. And what is happening here, it depends. You have to decide if your samples are coming from the place where non-bacterial DNA greatly outnumbers the bacterial DNA. So non-specific PCR amplification can occur. And in that situation, it is very useful to check a subset of your sequencing to make sure that matches to 16S reference sequences. So the pre-processing of the data for quality check is quite important for processing the metagenomic data. So I will quickly take the data analysis workflow because I would just want to tell you where the artificial intelligence application has improved this data analysis. So after pre-processing, we reach to OTU or ASV picking. And in OTU or ASV picking, what we do is we generate clusters basically. From the sequences, we classify them into different clusters. And in the taxonomic assignment, we use previously available data, the labeled data, and we give names to these clusters, right? This is what we do in basic data analysis workflow in bioinformatics. And we have several problems when you're doing this. Actually, you would have realized that things are not that simple as they appear to here. And I will explain you again that with the help of one example. And the same thing can be done by sequence alignment also, and we draw a phylogenetic tree with this, and then we label the branches, and we know which OTU or which ASV is present and what is the abundance of that. So we could do this by making a phylogenetic tree also. And for taxonomic assignment, we have many choices. We could use BLAST, and we could use machine learning algorithms also, particularly the RDP classifier is quite popular and gives you accurate results. So before we go further, we must understand what is the difference between OTUs and ASVs, right? So OTU, everybody knows, it refers to the operating taxonomic unit. And it is basically the consensus sequence for a particular cluster. And it has to have a similarity threshold of 97%. One OTU refers to one unit organism, and all the individual in an OTU has same role or function in the community. So in terms of biology, we treat OTU as one organism, unit organism. And in comparison to OTU, what is ASV? ASV is amplicon sequence variant. The ASVs, on the other hand, infer the real sample sequence variant within a sample. They are true observed amplicon sequence fragments from your sample. So I would just repeat, I said OTU comes from the threshold similarity of 97%. So OTU is perfectly computer based, meaning it will not use its mind. There's no intelligence involved in OTU determination. It will just simply determine that the threshold of 97% is reached, meaning it is different, right? And because of that, the OTU gives you error, is very much prone to errors. On the other hand, ASVs use artificial intelligence, machine learning. And what happens in ASV? they attempt to avoid arbitrary dissimilarity threshold, and they can distinguish sequence variants differing as little as one nucleotide. And they use machine learning algorithms, and they actually pick up the actual variant rather than the sequencing errors or because of the amplification errors. And the data 2 is a machine learning based division amplicon deionizing algorithm. And let me just show you what happens when we apply this algorithm rather than the normal OTU partitioning. So I assume that I have a community data where I have 4 type of sequences present. I have represented them with different colors here. So it is one community where we have 4 types, 4 colors. We isolated its DNA sequences, and we have 4 type of sequences present here, right? And we believe that, and it happens that we have errors in these sequences. I have highlighted these errors, though they are different, but they're going to merge with these, right? So it has errors. The yellow highlighted region shows it here, the error. And for the metagenomic analysis of this data, what we'll do is we'll do pairwise alignment. We'll do the pairwise alignment. It separates it into 3 groups, and we do clustering. And based on 97% similarity, like the typical OTUs, what it will do is it will cluster them into 3 main groups. And because of these errors, which were just simple, what do you call as sequencing errors or amplification errors, this took this green, which is a different, this which is coming from different organism, together with black, because this these 2 errors, they reduce the distance, and so they got clubbed together. And what happens is when we are picking up the reference or consequence sequence, it didn't pick any sequence for for the green one, so it missed green. And we reached where we could, this data will show you only 3 OTUs present in this community, right? So it shows you in OTU clustering, it will show you 3 uh, only 3 diversity of a 3, and the abundance would also be not be right. So the same thing when we do it with ASV identification using the Spirit. eSpirit is a machine learning based application, and when we do with that one, it does the deionizing, which I told you just now. And right, so what will happen? The same data, same things. We had 4 in this community. We have 4 uh, 4 type of OTUs or organisms present hypothetically, and we had errors in the sequencing. We did pairwise comparison like we did earlier. And what happens is with this pairwise comparison, right, sorry, what happens? It is learning errors through iteration. This will not accept this, and the algorithm will keep reiterating this thing. And if this error is found several times, then only it will assume as an error. So while deciding the correct consensus sequence, it will take into consideration, and it will be able to identify the errors based on learning through iteration. And after that, what we get the correction, and we get the complete. Correctly, we could know that we have 4 type of organisms present in this community. So that with that, I hope I could convince you that these machine learning algorithms, they really help us rather than the simple calculation, statistical calculation. When we apply them, that takes the system to to a better accurate and more applicable. So that was about metagenomic data, and I was just trying to tell you how the machine learning methods have improved our performance and system performance. And I tell you, the machine learning algorithms or applications are used at every step. I gave you one example of error fixing, but if you, I have table, I couldn't show you the complete table here because of this resolution problem. This reference I have taken from this paper, and if you see that, you see, say for clustering all types of machine learning algorithms, and at every step they've been used in metagenomic data analysis. So metagenomic data analysis for symbionts is taking full advantage of these algorithms, and they are getting refined every time, and we hope for more applications there. Then coming to the image data, wherein image data and how the machine learning algorithms are being applied is again to the end-to-end cycle, what is being done with the images which we feed, like the my spore images which I told you. We will give the imagery access, and what will happen is they will take in the data, it will create a training data, meaning and then after training it will make an in the test data it will make an inference that what does this image contain, and then it will give us an output, and we could based on this output we could do whatever function we wanted to do with this image. And in remote sensing image data, machine learning tools are used extensively, and we'll be specially interested in classification of images, which has found application, and in the clustering, prediction, deep learning, all are applied. But we study this with the help of two examples, two case studies. One is the lichen distribution. So lichen is symbiosis. So what is happening in lichen distribution? You know, lichen is a symbiotic association between fungi and algae. Lichen is a symbiotic association between fungi and algae. Lichen exhibit a higher range of visible reflectance and a more gradual increase in reflectance from red to near infrared than green vegetation. That is how in the remote sensing images, we could distinguish lichens from the other green vegetation or maybe the ground area. And instead of, once we have this sort of possibility, instead of manually visiting Antarctica and inspecting, we could use imagery to automate the distribution studies. Here is one picture which I have taken from this paper. It shows you the distribution of lichens in Antarctica, in tundra region. It's Project Tundra. So what artificial intelligence or ML is doing here is that it improves the prediction of the lichen distribution based on lichen distribution in this remote sensing data, and of the machine learning methods, the method which has specially found its application is neural networks. So using neural networks, quite accurate predictions about the lichen occurrence has been made, and it has been tried to automatize this using machine learning methods. Then another case study is of coral reefs biodiversity study. Coral reef is an underground ecosystem characterized by reef building corals, and reefs are formed of colonies of coral polyps held together by calcium carbonate. And corals, it's a marine invertebrate belonging to the class Anthozoa. Polyps are roughly cylindrical in shape and elongated at the axis of a vase-shaped body. They are also called as rainforest of sea because they cater huge diversity of microorganisms. How we study these corals in the marine environment? By manual photography by the divers, and CV methods, the computer vision methods have been employed for image classification, which we get from these divers, and making predictions about the habitats and studying the abundance and distribution of species in the reef ecosystem. The most common measurement of species abundance in in coral reef is percent cover. So with the photographs, we know the percent cover occupied by a taxa in an aerial view. And then we annotate randomly the points in this picture, in this image, and determine the relative frequency of various classes. And different classifiers have been used for this purpose, SVMs, KNN, or maybe decision tree to segregate images into different functional groups based on texture, color, and shape features. And SVMs have been found to be the best performing classifier, which gives accuracy greater than 80%. And in other studies, CNN have been reported to be very promising in classification of the coral reef images. And monitoring them also is equally important. Why? Because they are very good indicators of climate change, ocean health, and ocean acidification. And automated vehicles now are available with which we can monitor them automatically. And we have a continuous progress being made to develop automated image annotation. So AI methods are used for automation of these image methods. And automated image analysis from these automated vehicles is possible based on CNN methods. They've been found to be quite accurate. And they use training sets to learn an object or substrate of interest. And these automated image analysis methods are already to the point where they assess coral cover as well as more consistently than human observers. So with that, I come to an end of this talk. And I will just quickly summarize what we talked about today is like Alexa in our daily routine, we have AI tools being developed for helping studies of symbiotic organisms. We have AI methods like ML algorithms. They are applied at every step of metagenomic data analysis and biodiversity determination. We have CV, that is computer vision, is progressing towards the automation of coral reef monitoring in marine ecosystem and in remote sensing data and studying the lichen distribution, automatizing that. And a rapid progress is on the way. So if you want to learn more, we have, I have few references, you can learn from there, and might note that one of these references, my book, which I wrote some time back, and it's a good book if you want to learn the basics of bioinformatics, how to apply this and where to apply this. So I would just thank you for your patient listening, and I can stop sharing now. So I hope like it made sense to bring in between, but I thought it would be interesting to know how artificial intelligence is being applied in our symbiotic organisms. Thank you.
Thank you very much, Manju.
Thank you.
We really enjoy the talk. It was quite different from everything we have learned until now.
Yeah.
And I particularly enjoyed the part of the metagenomics data. So thank you very much.
Thank you. Yeah. Earlier I was planning to give on arbuscular mycorrhiza. Then I thought because we are writing a project on this, so I thought it would be better, you know, to do something different.
It was quite different. Thank you.
Yeah.
I think it didn't overlap.
I really enjoyed it because I didn't know about lichen, so it was very interesting. Thank you.
Thank you, Silvana. If anybody wants to ask any questions, I would be very glad to answer those.
Yes, I would like to make a question.
Sure. Yeah.
Thank you for the talk, as
Yes,
it
thank you.
was already said. Actually, your last slide was basically my question. If you would, if one would like to start learning machine learning, where should the person start with? Like, where would you recommend the person to start?
Yeah, so it all depends on your background. If you are a biologist, maybe, then like we do, we basically focus on the application part, meaning The algorithms are prepared by like my collaborators. They are in Carnegie Mellon University, USA. So they adjust my algorithms wherever, whatever I need. But once the algorithm is developed by them, and when it comes to the application part, say for example, metagenomic data analysis. So you pick up any data, the data is available in all these things, and then you can practice. And once you understand how it is functioning, then meaning it all depends on your background. If you are, because this, I tell you, this artificial intelligence or this bioinformatics, it's a multidisciplinary science. You can never be complete in this, because if you are from computer science background, you would miss all biology, right? So I think as a biologist, we could do this for more for application part, that, and we can help them to design for ourselves, but we should be familiar with our data, and application part is the forte where we could focus on as a biologist, I would say.
Yes, yeah, in my case, I was thinking about environmental DNA analysis, microbiome,
Yeah.
basically what you showed in the presentation. Yeah.
Yeah, and it is really going to work there, because I just, if you remember the slide which I showed, where with the help of machine learning, we could actually, OTU was not working fine, but we could shift it to ASVs, just because the machine could distinguish that this is an error, and we could do deionizing with that one. So these are the algorithms, and every day, because new algorithms come, but of course it is very promising, and you can never be in all the fields, but at least this is being developed for us. So maybe you can start with application part, I would say.
Yes, thank you.
Thank you. Anyone else wants to ask anything? It was a pleasure for me to interacting with you. At University of Lisboa has been, very favorite of mine. After 10 years of staying in India, I started traveling abroad in 2015 with Lisbon. I went Lisbon after a very long gap of travel. Yeah, please.
Hi. Thank you for the super comprehensive talk. I just had a technical question about how do you test for accuracy when you do like metagenomic data classification? Do you like always run a control with known OTUs or ASV composition, or do you just know the accuracy for the method itself? How does it work?
No, as I told you just now, we need to have a labeled data already present. So to make it successful, it has few steps. Like first, we should have accurate data isolation, sequence isolation. After that, we first have to be very careful about the pre-processing, so that we don't include the errors of the sequencing. And after that, we have this, you can use this chime software. With that, you'll be able to classify, or maybe you can do it with your own BLAST, and then sequence alignment, and then making a phylogenetic tree. See, I tell you, for example, of my mycorrhizal sequences. If I have a metagenomic data, I have a metagenomic data of my sequences. For one spore, we isolated the metagenomic data, and we isolated for all the, meaning AMF spore is associated with several bacteria often, isn't it? So we isolated them, but we need to have a database for comparison, because the taxonomic assignment is based on the labeled data. So these becomes the prerequisite. We have to have proper sequencing methods, so we don't include errors. Then we have to have good pre-processing, so that we remove the errors if they have come. Then we have to have proper algorithms which actually give us correct OTU or taxa assignment. Then we have to have pre-developed databases or labeled data with which we'll compare and give a name to this taxonomic taxa which we have obtained. And then we analyze this data, because with this we get the abundance data as well. Apart from diversity, we'll get the abundance data, and we will analyze it by several statistical methods. I hope this is what you were asking.
Yes, more or less. Maybe I misunderstood the method, but I was just more worried about when you, for example, when you don't know the species composition or OTU's composition, maybe they're not in your library either. How do you?
No, yeah. See, I tell you again. I will tell you what. See, no problems, please. What is happening is, remember, I had this tree. Do I do this slideshow again? Yeah, I can do that again. So I was talking of this. Yeah, I was here, not here. So let's do it this with this. Yeah. So you are saying so yeah, here, I just wanted to tell you this. See what happens in this artificial intelligence or general statistical analysis. So we are here after pre-processing, we have maybe 100 type of organism in one environmental data, right? So how will we know we have 100 types is we have clustering methods, meaning they will randomly cluster them into different groups. This represent one type, two type, 3, 4, 5. So these are the clustering methods. After we have developed clusters, we take one sequence from this cluster and we'll assume that as a consensus sequence. And then we will run it for similarity in the already existing databases, right? And this is taxonomic assignment. So we will give name to this cluster after this, and several unidentified clusters always remains because we don't have labeled data available for that. Yeah, this is what you were asking.
Yes, thank you.
Yeah.
Sorry, can I ask a question?
Yeah, please.
How can we contribute to those data image? For example, I do scuba diving and I like to take pictures. I like insects. I like to take pictures. I used to contribute to the lists. I don't know if you know, it's a huge database here in Portugal for all kind of animals and plants.
Yeah.
But for those databases, can we contribute and how?
Yeah, I think you can, but we have Simon working on this coral reefs and this thing, but we have to be very focused, and you have to focus on one particular type of habitat if you're going to make them useful, right? And maybe because you are a photographer, you would know, but there are a lot of databases existing. I can cite some, not now, I can send you later that maybe you can deposit your images if you want. Anyway.
Okay.
Then.
Thank you.
Yeah, thank you. So good to see you all there at Lisbon and doing good work. Yeah, Silvana? Yeah.
I can understand that in the article for the Trunda it's quite easy because you mostly have only lichens and mosses. But if you are talking about a forest with a lot of different layers that is stratified, there are methods also to identify the different layers, or for the moment it's impossible?
I think with images I think they are doing it with limited success, but not too sure. Right now I was focusing on lichens, and we really didn't bother about the lichens which grow on trees and this thing. We just focused on three aspects: land, lichens, and trees. So, but I saw papers where mycorrhiza, actomycorrhiza also is being tried to be distinguished. And I did see papers where they actually identify which tree they are talking. So of course they are getting success. So, but here, because I was just focusing on the artificial intelligence methods where they have got success and they are helping in automatizing the things, but things are working. I'm quite hopeful that things are working.
Thank you.
Thank you.
Okay, so one last question. Okay. Manju, we will have Eric after you, so if you want to stay with us,
Yeah,
please.
I will try to stay for a while. Thank you very much. Thank you, Silvana, for this opportunity that I could see your students and talk to them. It was a good change. I felt that I've traveled to Lisbon once again. Thank you.
Thank you very much.
Thank you. So I'm waiting for to see Eric.
Sorry?
Yeah, has he come?
No, I don't think he's here yet, right?
No, seems to me it's not here. We'll, okay, wait a couple of minutes.
Yeah, we can go to the bathroom or whatever, and
Yeah.
we can be back.
2 minutes break.
Okay.